Hi, this is MEDMASTER. In this video, we're gonna talk about Krebs cycle or citric acid cycle. In the previous video, we talked about glycolysis. We said that glycolysis is a series of enzymatic reaction which converts a molecule of glucose into two molecules of pyruvate. During this reaction, we produce 2 net ATP and 2 NADH. If there is a good supply of oxygen and in the presence of mitochondria, these molecules of pyruvate will get in the mitochondria. So now let's zoom in and see what is going on inside the mitochondria. Inside the mitochondria, these molecules of pyruvate will be converted to acetyl-CoA by the enzyme that is called Pyruvate Dehydrogenase complex. Pyruvate, it has 3 carbon, and acetyl-CoA, it has just 2 carbon. So that means we lost a carbon in the form of CO2. Then we say that dehydrogenase, this enzyme is called pyruvate dehydrogenase. Whenever you hear about dehydrogenase, of course, we gotta reduce NAD to NADH. Then the synthesized molecules of acetyl-CoA will react with a 4 carbon molecule that is called oxaloacetate. Then oxaloacetate plus acetyl-CoA will produce a 6 carbon molecule that is called citrate. This reaction is catalyzed by the enzyme that is called citrate synthase. Citrate synthase, it catalyzes irreversible reaction. Irreversible reaction. So this enzyme is highly regulated. If there is a lot of citrate, a lot of ATP and a lot of NADH and also a lot of succinyl-CoA, all these, they're gonna inhibit these enzymes, but these enzymes will be activated by ADP. So because of our goal is to produce ATP, because of that ATP will inhibit citrate synthase, and also NADH inhibits citrate synthase because a lot of NADH will be oxidized in the electron transport chain to produce more ATP. Then citrate will be converted to isocitrate by the enzymes that is called aconitase. This enzyme aconitase is really sensible to rat poison. We know that the rat poison contains fluoroacetate. When fluoroacetate binds to aconitase, that will inhibit the conversion of citrate to isocitrate. Then isocitrate will be converted to alpha-ketoglutarate. So isocitrate it has 6 carbon, but alpha-ketoglutarate it has 5 carbon. So that means we lost a carbon in the form of CO2. So what is the name of the enzymes which convert isocitrate to alpha-ketoglutarate? This enzyme is called isocitrate dehydrogenase. We say that we never hear about dehydrogenase. Of course, we gotta reduce NAD to NADH. As these enzymes, it catalyzes irreversible reaction, it is also highly regulated. So calcium and ADP will activate these enzymes. Calcium and ADP will activate these enzymes, but a lot of ATP will inhibit these enzymes. Then alpha-ketoglutarate will be converted to succinyl-CoA. So let's count the carbon of alpha-ketoglutarate and the carbon of succinyl-CoA. Alpha-ketoglutarate, it has 5 carbon, and succinyl-CoA, it has just 4 carbon. So that means we also lost a carbon in the form of CO2. So what is the name of the enzymes which convert alpha-ketoglutarate to succinyl-CoA? This enzyme is called alpha-ketoglutarate dehydrogenase complex. We say that whenever you hear about dehydrogenase, of course, we're gonna reduce NAD to NADH. Then as alpha-ketoglutarate dehydrogenase complex, it also catalyzes irreversible reaction. These enzymes also it is highly regulated. These enzymes can be inhibited by NADH and ATP. If there is a lot of NADH and a lot of ATP will inhibit these enzymes, but calcium will activate these enzymes. Okay, then succinyl CoA will be converted to succinate by the enzyme that is called succinate thiokinase. In the previous video we said that kinase will add phosphate. Okay, so here succinate will remove the CoA from the succinyl CoA and it will add inorganic phosphate to GDP. So GDP plus inorganic phosphate, we're gonna produce GTP. Okay, remember that GTP is equal 1 ATP. Okay, then succinate will be converted to fumarate by the enzymes that is called succinate dehydrogenase. But here we're not going to reduce NAD, but we are going to reduce FAD to FADH2. Then fumarate will be converted to malate by the enzyme that is called fumarase. Okay, then malate will be converted to oxaloacetate by the enzymes that is called malate dehydrogenase. Of course, here we're gonna reduce NAD to NADH. Okay, then now let's count what we produce during the process of Krebs cycle or during the cycle. So let's count the number of NADH we have. Okay, here we have 1 NADH, 2 and 3. We produce 2 NADH per cycle. And then let's count the number of GTP or ATP. We have just 1 GTP and we have 1 FADH. Okay, and we have here we have 1 CO2 and here we have it become 2 CO2. So that means we produce 3 NADH, 1 GTP, 1 FADH and 2 CO2. Okay, I hope you enjoyed the video. If you liked the video, please subscribe to our channel and see you next time.